Thank you for the introduction. It's really nice to be able to be here and present with you to you today. I'm going to talk to you today about what we consider to be next-generation manufacturing technologies for cell and gene therapies. So I'm sure you're all very familiar in the space with some of the traditional approaches using plasmids. I'm not going to talk about those technologies today. I'm going to be talking about completely different alternatives that we've been working on for a number of years. So just very briefly about the company. So WuXi Advanced Therapies is one of the divisions of WuXi AppTec, and it's primarily based in the US, where most of the employees are. I've got about 400,000 square feet over there with 30 different GMP suites. I think it's fair to say the majority of the work that goes on over there is GMP. Last year, WuXi Advanced Therapies acquired Oxgene, which was a company I founded 10 years ago. We are operating as their research facility, essentially, so developing new manufacturing platforms, doing discovery work for clients as well. And then in China, we do a lot of plasmid manufacturing at GMP, as well as also viral vector manufacturing. One of the things that the Philadelphia site is most well known for is cell therapy manufacturing. They've released close to 2,500 cell therapy manufacturing lots, so that's one of the main things that they have historically done. The Oxgene side, the majority of work that we've done is on the viral vector piece, so primarily AAV and Lenti. And what we're really trying to build as a company is this continuum of services and solutions that we can then work with clients all the way from the very early concept, so from the DNA stage, all the way through to BLA and commercial supply. So the early bits in the top that you can see where we do things like promoter discovery, capsid discovery, vector engineering, cell line engineering, that all happens in Oxford in the UK, which is where I'm based. And then once something is working and we've demonstrated it at smaller scale, that then passes over to China or the US, depending on where the client wants to work. And that goes first to the development teams, then to the banking teams, and then subsequently on to all the viral vector manufacturing, cell therapy manufacturing, plasmid manufacturing, depending on what the client needs. So the first platform I'm going to talk to you about is our stable lentiviral manufacturing cell line. So I'm sure as many of you are aware, when you traditionally make lentiviral vectors, you would transfect 4 plasmids into cells. Some people have historically done it in adherent cells. A lot of companies now have transitioned over to suspension cells. But the ultimate goal for a long time has been to try and make fully stable producer cell lines for lentiviral vectors. For retroviral vectors, they've been around for probably 25 years. So in the mid-90s, they started to produce retroviral vector producer cell lines. But nobody has ever really cracked it for lentiviral manufacturing. And for that reason, the majority of lentiviruses are still manufactured using quadruple plasmid transfection. So the principle is relatively simple. You integrate the genes, and this is the diagram on the left, you integrate the genes involved in the manufacture of lentiviral vectors into cells. Some of these genes are quite toxic to cells, so they have to always be done in some form of inducible expression. So pretty much every system uses an inducible expression system for both VSVG and GAG-POL. And then the way that we have done it is that we have initially started by making packaging cell lines, and then we come in a second step and introduce the customer's specific vector into those cells to produce stable producer cell lines. The benefits of doing that are that you obviously don't need to manufacture plasmids anymore. The reason that that's important is probably in the region of 40 to 45% of the cost of manufacturing lentiviruses are down to the plasmids and the reagents associated with the plasmids and the reagents are involved in transfection. So it's a very large portion of the manufacturing costs of lentiviral manufacture. The other issue is that when you want to transfect cells, you have to do it at a relatively low cell density. So if we wanted to move the industry to something more akin to monoclonal antibody manufacture, where you would significantly increase the density of your cells in your bioreactor and then start to produce your material, you simply can't do that with lentiviral plasmid transfection because you have to transfect at a relatively low cell density. So for 2 reasons. One, it is significantly cheaper, but secondly, it also opens up a lot of additional parameters that you can mess around with and tweak with in the bioreactors that you simply can't do when you're using plasmid transfection. So this is the only piece of data I'm going to show on this, and this is really the culmination of about 5 years of hard work. So the data on the right here is just 3 clones that are fully stable producers for lentiviral production. They're producing in the low 1 × 10⁸ infectious units per mil, which is very good yield and comparable to our fully transient system that we've been also developing for a number of years. The process is relatively simple. As I've alluded to, we have a packaging cell line. We take the specific vector of interest to the client, we integrate that into the cells, and then we go through a very large automated screening process using robotics to identify individual clones that are producing high levels of lentivirus when we induce them with doxycycline. These titers have been titrated on HEC293 T cells. If you did them on something like sub-T1s, you might see slightly lower levels. And of course, we're not trying to save jellyfish by making GFP. What we ultimately want to be doing is something therapeutic. So we are in the process of constructing CD19 CAR equivalents just to demonstrate something that is therapeutically relevant. We have got to the pool stage with those constructs. Those are currently yielding around 4 times 10 to the 7, which is similar to what this was producing actually at the pool stage. And we should have data from those clones probably in the next 2 weeks, hopefully in time for the ASGCT in Washington in early May. So that's the only piece of data I'm going to present on that, but the thing I will highlight is that these vials are available to people if they want to evaluate them in-house. So if people do want to validate the data that we're getting, we do external evaluations all the time. And these vials have now been banked as a research cell bank that people can access to test out themselves. So switching from lentivirus to AAV, we've been working on AAV for a number of years. We tried to initially a similar approach to what we did with Lenti to make stable cell lines. We found it very difficult. We screened in the region of 2,000 clones and ended up with basically nothing at the end. But we were parallel tracking a completely alternative technology which uses a modified adenovirus system. And I'm just going to briefly talk about how that system works and some of the data we've generated using it. So at the moment, similar to Lenti, most of AAV is being manufactured by plasmid transfection, in this case a triple plasmid transfection. However, that's not what happens in nature. So in nature with AAV, what happens is the virus will replicate when another virus comes into that same cell. So pretty much everybody knows that that's typically adenoviruses or herpes viruses. And it wasn't always the case that AAV was manufactured primarily with plasmids. In the early 2000s, the majority of it was actually being manufactured with wild-type adenovirus. Those processes were very efficient. They were actually scaled up to over 2,000 liters, producing very highly pure AAV, or at least very highly packaged in terms of percentage full AAV, with very good properties in terms of how infectious those AAV particles were. But the industry has, for very logical reasons, transitioned over to plasmids. The main reason is because people were using wild-type adenovirus. So they were actually using the wild-type infectious adenovirus that we use, that is the pathogen that we've all heard of, in a HeLa cell system. And HeLas contain HPV DNA as well, so they're not preferable from a regulatory perspective. So for 2 reasons, people didn't like using that platform. The other main reason was that when you manufactured AAV with adenovirus, you got pretty much the same amount of adenovirus as you ended up with AAV. So you then had to purify all of your AAV away from the adenovirus, purify your AAV away from the contaminating adenovirus. And so what we wanted to do was to see if we could solve this through a genetic switch that we could build into the adenovirus. And this work was published in Nature Communications last month. And the principle behind the technology is relatively simple. So as humans, we've divided the adenovirus life cycle into 2 convenient stages. There's the early phase, which both adenovirus and AAV require. Those genes are the ones you would recognize from the helper plasmid that we typically transfect into cells when we're doing triple plasmid transfection. The late part of the adenovirus life cycle is completely useless for AAV. It essentially all it does is produce structural proteins that subsequently produce adenovirus, which contaminates our AAV preparation. So what we'd ideally like to do is have an adenoviral vector that we can put into cells that will then express all of those early genes, but if we could switch off all of the late genes, we would then be able to prevent the adenovirus from contaminating our AAV preparation. And that's essentially what Tesa technology is. So Tesa stands for tetracycline-enabled self-silencing adenovirus. And the principle is relatively simple, although it was an absolute nightmare to make it. It essentially incorporates 2 different modifications. So firstly, what we've done is we have introduced a repressor binding site into the major late promoter. Now, this is the promoter that drives all of these structural proteins, which subsequently produce this adenovirus particle. What we then did is we actually put the repressor that will bind to that binding site downstream of the promoter. So what that means is the more this promoter tries to produce these structural proteins, the more it actually represses itself. So you get a negative feedback loop built into the virus, which means no matter how hard it tries to produce itself, it simply cannot because it represses itself even more. And we've shown, and in that paper you'll see that the regulation is exceptionally tight. It's in the order of 8 log reduction in the crude supernatant of the amount of adenovirus that will come out of these cells in the absence of doxycycline. Now, what's great about the system is that when we want to produce this adenovirus, because we will need to do that if we're going to use it to manufacture AAV, we just do it in the presence of doxycycline. So when we're manufacturing the adenovirus, we add doxycycline. It's as if it's a normal wild-type adenovirus because this protein can no longer bind to this promoter. When we want to make AAV, we simply leave out the doxycycline. We get all of the early gene expression, which is the helper functionality that we need for AAV manufacture. We get genome amplification, and hopefully we get AAV coming back out of the cells. And so what we've done is we've gone through a number of different iterations, and this is one of the versions that we're working with at the moment, where we have 2 versions of this Tesa system, one encoding the AAV genome, the other one encoding AAV rep and AAV cap. What we do is we introduce both of those viral vectors into the cells, and subsequently we only get AAV coming back out. Now, this is just showing some data here for a therapeutic transgene, so it's not GFP. This is a therapeutically relevant product. And what you can see in suspension, serum-free process, in this particular instance for AAV9, we actually managed to get above 1 × 10⁻12 GCs per mil, which is very, very high levels of AAV. The reason for this is in part because these viral vectors will get into every cell within your population. So it's not like transfection where a portion of the cells won't get anything. Another subportion will get a certain amount of helper and the rep cap and the genome plasmid. Everything basically gets all of the components that are required. So we're getting very high productivity in this case of AAV9. This is showing data where we've done a head-to-head comparison between the plasmid-based approach, and this is a standard plasmid system, and compared it for RepCap1, so AAV1, AAV2, 4, and 5. And you can see varying degrees of improvements that we've seen, and this is, again, all in suspension-based process. This is showing cells GC per cell. You can see here for some of these serotypes, we're getting above 1 million particles per cell, which is very, very high productivities for AAV. This is showing similar data for RepCap6, for RepCap7, and RepCap8. What we're also showing here is that we've managed to double the percentage of packaging efficiency. Now, what I will say here is that this is a cell line that WuXi had originally developed. When we were acquired, we then inherited a cell line from WuXi Advanced Therapies, a cell line that is considerably better. And actually, as you'll see in a second, the data is even better when we've scaled it up to 50 liter. Now, another thing that's important to highlight is how infectious the AAV particles are. So one of the reasons we wanted to look at using adenovirus is because it's what happens in nature. And so what we wanted to then determine is whether those AAV particles are able to get into cells more efficiently. And what we find is that for a number of different serotypes, when we compare how infectious the AAV is and its ability to get into cells, and this is just looking at GFP, we can see that the particle quality is significantly improved for a number of different serotypes. We then wondered whether it was our helper-free system. Maybe that wasn't very good. So what we did is we bought reference standards for AAV6, for AAV8, and AAV9, and then did a TCID50 assay to determine how infectious those AAV particles are. And essentially, the lower the ratio, the more infectious the AAV particles are. And what we've seen is above a 10-fold increase for a number of different serotypes that we've worked with. So it does appear that the particles are being better assembled than when we use the plasmid-based system. Now, we've now gone on to scale this technology up to 50 liters. There's actually a 200-liter bioreactor running that's right now for AAV2. This is just showing where we had a 1-liter bioreactor plasmid control at the same time and showing we've got 10 times more for AAV6 when we're using the 50-liter bioreactor. This is just showing what I was alluding to earlier, that pretty much every cell gets this adenoviral vector introduced into it. You get very high transduction levels in the bioreactor. It's also relatively independent of cell density, and this is using the same cell density that you would use for transfection. So we know we can go a lot higher than this, potentially even 10 times higher than this, if we can get the bioreactor and media conditions correct. In terms of overall yield, it was close to 4 × 10⁻16 AAV particles for a single 50-liter bioreactor. Just looking at what the particles look like. So this is, as I was saying earlier, we were seeing maybe around 20–25% particle encapsulation in the earlier data. In this particular 50-liter run, we got 66% of the particles in the crude material that contained the AAV genome. I think that might be a slight overestimation because when we go to the next stage, it dropped down to about 55%. But nonetheless, by the time we purified the material, by ELISA at least, it was showing at 100%. On the next slide, you'll see it's ever so slightly below that, but not far off. We also looked at how infectious those particles were relative to the number of total GC. What we found was around 3% of those AAV6 particles were able to infect cells. This is in a HeLa model, so obviously not a target tissue where we've titrated the AAV, but nonetheless significantly higher than what we would see with the standard plasmid-based approach in the same GMP manufacturing context. This is just showing by analytical ultracentrifugation what that material looks like. At the end, we were at 94.5% full AAV6 particles, which is significantly higher than we've ever seen for any of the other approaches we've been working with. So very, very pleased to see those results. So just to conclude on the lentiviral stable cell line, so we've generated fully stable eGFP producers, and we have the CD19 CAR data that's hopefully going to be available in the next couple of weeks. They are entirely independent of plasmid transfection. They do require doxycycline to induce them, so probably that lentiviral material is not suitable perhaps for an in vivo application, but certainly should be acceptable for an ex vivo application. And those vials are available for evaluation, and if you want to, you can email this email address here. And then Tesa technology, this is the actual number that the adenovirus is reduced by. If you look into the paper that I showed earlier, it has all of the data around that. And what we've shown is that for the Tesa, what we call Tesa 2.0, which is where we use 2 of these Tesa vectors to make AAV, It significantly increases AAV particle yields for all of the serotypes that we've worked with. It also improves particle quality for the ones where we have a half-decent in vitro assay, and it does remove entirely the dependency on transfection and the issues and limitations that that presents. And once again, those materials for Tesa are available for external evaluations. We can also do evaluations in-house, but if people want to actually just buy those adenoviral reagents and test them in their own laboratories, then they're welcome to do so. So thank you all for your time.